This black fly protocol can be used for recently trapped flies or those that have been stored in isopropanol. Be sure to rinse the flies in 95% ethanol to proceed. To rinse the flies, pour them into a sieve and rinse them thoroughly in 95% ethanol. Then allow them to dry, but do not allow them to desiccate. Gently transfer the black flies within each pool into a 15 ml conical tube. Then you may place the tubes in the freezer at -70 degrees Celsius or on dry ice for a minimum of 2 hours. When the black fly pools are frozen, you can snap the heads off by pounding the 15 ml tube on the bench top in a firm flicking motion, but not too hard so that you do not crack the tube. It is best to utilize a piece of foam or mat on the tabletop to help soften the blow and prevent cracking the tube. You can also work on several tubes at one time by placing them inside the fingers of a glove to pound multiple tubes at once. Collect the fly material by adding 95% ethanol to the tube, then pour the contents into a stacked sieve. Then wash with more ethanol to separate the heads, bodies, and legs. Collect the heads and bodies by washing them off of the sieves using 95% ethanol into a plastic pan or petri dish. You may further sort them using a transfer pipette. You may also use a dissecting microscope to separate the remaining heads and bodies using a transfer pipette. Place all black fly heads from a single pool into a 1.7 ml tube using a transfer pipette. You will need to remove ethanol as you continue to add heads, but leave 1 ml in the tube for storage purposes. When you are ready to proceed with homogenizing the fly pools, start by removing the ethanol in the 1.7 ml tube using a fine-tip transfer pipette. Carefully remove all the ethanol, leaving the heads in the tube. Allow the fly heads to dry briefly before proceeding with the next step. But do not allow them to desiccate. After removing the ethanol, you will want to add 200 microliters of DNA-RNA shield to each pool of fly heads. After this addition step, you may leave the pools at room temperature for up to 30 days before extracting DNA. Next, you will manually grind the fly pool using a disposable homogenizer, also known as a pellet pestle. Mechanically homogenize each fly pool after the addition of DNA-RNA shield using a single-use pellet pestle. With a grinding motion, be sure to thoroughly knead the fly heads into a well-blended mixture. Gently roll the pellet pestle along the sides of the tube when finished to remove fly debris. After homogenizing each fly pool, centrifuge the tubes in order to collect the homogenate supernatant. To do so, place the tubes in a microcentrifuge and spin for 6 to 8 minutes. Make sure to balance the centrifuge. And add an additional tube whenever you have an uneven number of samples. You may also repeat the centrifugation process when needed. Once the pool has been properly centrifuged, carefully remove the tubes without shaking the pelleted debris. Gently remove approximately 180 microliters of supernatant and transfer it to a new 2 ml screw cap microcentrifuge tube. Be sure to close the tube tightly. Then wrap the screw cap tube with parafilm for transport and storage. You will notice that the supernatant solution is tan or gray in color, but it should not contain any fly head debris. Any DNA contained within the homogenate supernatant will remain stable for up to 30 days at room temperature. You may also keep the specimens frozen for extended periods of time. Fly pool tip: for optimal retrieval of supernatant discussed in this protocol, it is best to create fly pools between 1 to 50 flies per pool. Be sure to label each one of the sample tubes with a unique identifier and keep a log of each pooled specimen for reference.